Now what we're going to do is have a go at extracting some DNA from fruit, or doing what's called a DNA extraction. DNA is a universal molecule, so all organisms have DNA within them. Doesn't matter what you eat or what you look at or what you touch, if it's a tree or if it's a sandwich or whatever it is, the molecules within them are going to have DNA, or the cells within them are going to have DNA. A fruit, just like any organism, isn't just made out of one cell, it's made out of millions of cells or trillions of cells. And those cells aren't just sitting by themselves, they're nice and controlled and kept in place by something called an extracellular matrix, which is a matrix of protein fibres, which surrounds all of the cells in a tissue, or in this case in the fruit, and it holds them all together. So what we'll do now is I'll take you through and I'll show you the bits we're going to use, and then we're going to have a go at doing it. Okay, let's jump over here. Okay, Maddie, so we're over here now at the setup, and we've got everything we need here to extract DNA from fruit. We've got our kiwi fruit, which we're going to use. Probably going to use one today, that should be fine. We've got what's called wool mix, or what I've called DNA extraction, where I've just taken some detergent, I've mixed it with some salt, okay, and that's going to help us to break down that cell membrane. We've got our meat tenderizer here. We've got our alcohol over here, which is chilling, that we're calling isopropanol, and a whole bunch of test tubes and things which we'll use along the way. So I'll get you to get started. The first thing we have to do is cut up our kiwi fruit into little bits. Having the skins on okay, because we're going to be straining it anyway, but I need you to take this one kiwi fruit, maybe cut the ends off, and then cut it up into slices, and then we need to place it in this Ziploc bag if we can. Excellent, that's fine. So just chuck it in that Ziploc bag then, don't do it up just yet. The first thing we want to do is break down that cell membrane, and there's so many cells that it takes quite a long time, so we're going to put that in first. So put 20 ml of the extraction solution into here, and then place that in that Ziploc bag, please. Yep. Is that about right?
Yep.
Excellent. Great. Now what we're going to do is try and remove as much air as possible. So push it down and then close the zip. So now what I'm going to do, Maddie, is try and get as much air out as you can, because we're going to smush it up. So push as much air out as you can, or I'll help you with it. And then what we'll do is just close up that Ziploc bag. You can mush it in your hands, and you want to get it really mushed up. So about 5 minutes worth of really smushing it up. Okay? We're trying to get that extraction fluid through as much as we can to break down all those cell membranes. So we need to get it as liquidy as we can. Right. So it's good fun, have a good play, spend some time with. Right, so we want to get all those little bits of kiwi fruit all nice and smushed up. So we get it as liquidy as we can. Because we really have to try and remove those cells from all of the tissues. And if we get big lumps in, then we're not going to get that solution all the way through the tissue. Like I said, the key to this process is temperature. We have to keep everything really, really cold. The solution, the alcohol, everything. So what we really have to do now, we need to give it time to do its work, the detergent, to remove all of those the cell membranes from all of the cells. So we're going to take this and we're going to place it in this ice bath. We're going to leave it there for about 5 minutes. All right, about every minute or so, we're going to take it out. We're just going to sort of gently push it around. Yep, that'll do. So we'll shove it down in there. Perfect. And we'll leave it in there, as I said, for about 5 minutes, just to let all that wool mix do its work and to let this whole thing cool down.
Okay.
All right. What we're going to do now is we're going to filter the contents, and then we're going to add in our protease so we can start removing those proteins, because all those lipids are broken down, all those fats, so we can get rid of the protein. So take that out of there. All right, we'll just leave that on the bench, that'll be fine. I'll get you to grab that Chuck's cloth. We're going to filter it into this measuring cylinder, so put that Chuck's cloth in there. What we're going to do is we're going to put all of that in here, and you and I are going to smush it through, and we're going to get that lovely liquid to come out. So yep, all of it in there. It's already starting to come out because we mushed it up nice and good. Okay, this is the fun messy bit. So place that on there, and you and I, I'll help you with this. Let's pick it up, and we're going to smush it through to get all that juice out. Right. Excellent, that's plenty. I'll put this in the sink, nice and clean. Now what we need to do is we need to place some of that into a test tube. So I'll get you to grab a test tube. Yep, like that. And it doesn't really matter how much. Let's go about very much. Okay. Okay. Now we need to get our meat tenderizer. All right. So grab that meat tenderizer, and we're going to remove all those proteins in your little spatula there. Excellent. And we don't need much. 1 or 2 little spoons will do. So use the other end, the little scoop end, and scoop out maybe 2 little salt grains. Yep, about that much. about another half one of those. Perfect. Chuck that in there. Excellent. Grab a stopper, put the stopper on there, and then just gently invert it a couple of times just to mix that through. A couple more times. Still a few bits in there, we'll get them all mixed up. Yep. That'll do fine. Back in there, we'll take that stopper off. And we're going to leave it for about 10 minutes, once again, because we need to make sure that all those proteins, that extracellular matrix, gets removed from all of that tissue that's in there, so that we can then remove the DNA.
Okay.
All right? Great. Okay, Maddie, so it's been about 10 minutes. Now we're right at the end of the real exciting bit where we can extract the DNA. So the DNA is now in here somewhere. It's in a soup. It's been released from the proteins, from the extracellular matrix, and it's been released from the cell membrane. So now what we can do is we can take it out. What we'll do is we'll get a nice clean test tube, because this one's a bit gunky. So grab a clean one. And I'll get you to pour in, once again, it doesn't really matter how much, probably about this much within the new test tube. Yep, that'd be perfect. Now what I want you to do is we're going to grab the alcohol, which is the isopropanol. All right, now it's a little bit tricky to get it to work. So we need to invert the test tube slightly. You'll open up that lid. Yep. You're going to invert the test tube slightly, however you want to do it. All right. Now you're going to run the alcohol down the side of the test tube, and you're going to squeeze about medium hard. You don't want to squirt all the stuff out, but you also want it to sort of slosh through a little bit. We're getting about 2 centimeters worth. So just keep going to it till you to stop. All right, off you go. Keep going, keep going, keep going. Harder, harder, harder. Keep going, keep going, keep going, keep going, keep going, keep going, keep going. That'll be fine. Excellent. So what you can see there, starting to separate out, right? Because the alcohol normally would be very clear, very pure. You can see that it's very misty. And what we've done is we've separated out the DNA from that solution. So now what we can do is we can have a go at spooling it. Right? So what you can do is you can place that skewer in there. Yep. And you'll see that you'll start, as you do, you'll be able to see it almost a solid that you'll start to move. There it is. Okay, and you can start to pull out, and there it is. So that's our pure DNA. All right, so we can almost spin it like a bit of fairy floss, like this. Okay, and as we do, when we pull it, there we go. We can see our pure DNA, and that's all it is.
Yep.